This is the third video of cellular respiration as we are studying aerobic respiration. The first video is about glycolysis, the second one is about the Krebs cycle, and the third one is about this one, which is the electron transport chain. So let's start with the big picture here of cellular respiration. We started with glucose. It goes into the reaction of cellular respiration. We have already turned glucose into pyruvate and then acetyl-CoA, and then we have basically turned it then into FADH2 and NADH from the Krebs cycle. So we've already broken down glucose that goes in. We haven't used oxygen yet. We have given off carbon dioxide in the Krebs cycle, so that piece has been given off. Water hasn't quite come into play yet, and we have made energy. Let's recall that in glycolysis we net 2 ATP, so we give off 2 ATP total. In the Krebs cycle we also gave off 2 ATP, so now we've given off 4 ATP. But we're going down this road and we need to give a lot more ATP. So we're on the road of aerobic respiration, which means with the presence of oxygen. So here's glycolysis, net 2 ATP going in, 4 coming out, to net 2. After glycolysis, we turn glucose into pyruvate. It, through a series of reactions, becomes acetyl-CoA. Acetyl-CoA goes into the Krebs cycle. And remember, 4 things come out. Carbon dioxide is waste. We labeled that. NADH, we starred NADH and FADH2. We starred those because those things, the goal products of the Krebs cycle, and ATP. So we also got 2 ATP from the Krebs cycle. We studied after the Krebs cycle, we kind of put everything together. We started with glucose, went to pyruvate, NADH, FADH2, and 2 ATP. With glycolysis here, with the Krebs cycle here. But we have to remember the goal of cellular respiration, and that was to produce ATP, adenosine triphosphate. And that's going to be through this right here. Now I'm going to do something a tiny bit different, and that is to add things as we need them, because this picture is already complex. So this is the electron transport chain, ETC, in the mitochondria. So let's do some anatomy of the mitochondria first. We have an outer membrane, which surrounds the mitochondria's inner membrane, which is all squished up, remember? So we have outer membrane, inner membrane, which has folds in it called cristae. And those are CRISTAE. So those are here, the folds in the membrane. Now the reactions occur in these folds, and that's where we are. This membrane here is this membrane here, these folds. Deep inside the mitochondria we call in the matrix. So I want you to label this picture first, which is outside of the inner mitochondrial membrane, which would be this space here, and then this would be inside. We call this the matrix. So we're going to label the outside of the inner membrane and then the inside, which is called the matrix. It's that blue stuff, okay? So this is the blue area here. Alright, so we're going to add some steps. First, electrons from NADH and FADH2 pass through the inner mitochondrial membrane. So those were made in the Krebs cycle. Here's NADH coming through, and I'm going to go ahead and add this to be step A. So you can do that now. So NADH is broken down. The electron is grabbed from the NADH, also from FADH2. Now that's not in this picture, but we're going to add it because we're so smart. So FADH2 and NADH lose electrons and they become the electron transport chain. That's why we need these two molecules from where? The Krebs cycle. This is where they're made, and so then they're broken down here. Remember, we're in the mitochondria. Never forget where we are. So that's letter A. So we started electrons out here, and this is the action that will start the whole thing. In letter B, we have hydrogen ions being pumped out of the membrane as electrons pass through the pump. And does that sound familiar? There is another place where we have electrons passing through, and as they pass through, they push the pump, they push the button, they give the pump energy to work. So this is step B, and really this is happening in a couple different areas here, but we'll label it right here. We're going to grab this hydrogen and we're going to pump it out, and the electrons cause the pump to work. Now, is this active or passive? Well, it's going from an area of low concentration to an area of high concentration. So, for hydrogen ions, so it's active. Okay? So that's step B, we're pumping hydrogen out. In step C, there are a lot of hydrogen ions outside of the membrane and only a few inside the matrix. And so we have a concentration gradient. Now, if you think about photosynthesis, I bet you can figure out what happens next. When we have this abundance of hydrogen in one place and very few in the other place, what happens is that they passively diffuse through. So let's label C, which basically is not really a step, but it's kind of the accumulation of hydrogen ions. I'll put them right in the middle of this. Or maybe I should put it by the high concentration so that you can relate that to the reading here. So there's a concentration gradient. What happens next is in letter D. So it's after the picture. We'll go on to letter D. The hydrogen ions diffuse back into the inner membrane through a carrier protein, which is ATP synthase, that adds a phosphate group to ADP, creating ATP. So this sounds just like photosynthesis, and it is just like photosynthesis. So this is letter D. Add it. I'll read it again. Hydrogen ions diffuse back in to the matrix, to the inner compartment of the mitochondria, through a carrier protein, ATP synthase, that adds a phosphate group. So if you look at the picture, this is labeled the ATP producing carrier protein. It's called ATP synthase. If you want to write that down, that would be fine. And the hydrogen, all these hydrogen accumulating, they're going to come through here passively from high to low, right? In. And as they do that, this protein makes ATP. In fact, it makes 34 ATP, not just a couple of ATP. So, let's finish. Letter E, the hydrogen ions and electrons combine with oxygen to form water. So it's like the opposite of photosynthesis, where we split water, remember way back? Well, now we have free hydrogen ions and electrons, and oxygen comes in to form water. So we need to show that as letter E. So here's oxygen, free hydrogen that have diffused in, and the electrons that come in through the electron transport chain. So we'll label this E. And we are making water. Now remember, water is a waste product of cellular respiration. And the goal is to make ATP, which we have done. All right. To sum up, glycolysis makes 2 ATP, the Krebs cycle makes 2, the electron transport chain actually makes 34 ATP for every glucose that gets broken down. And that's approximately, that's not a perfect number. It's an average for a total of 38 ATP. So this number is 38 ATP in aerobic respiration. So, long video, you did a great job. And then I will start another video for anaerobic respiration.